is the plastic we have considered that we have only the inhalation case in our study. So this is the very brief overview of my study. Before going to further, I just want to show you a video that has been considered the human airways, how the nanoplastic or any other particle inhale in our body. So this is the reason that normally considered in the fluid dynamics model, but we are going to talk about the molecular dynamics. Down this scale, we have actually worked on that. So this is the 18,000 of Lagrangian particles we have considered in case of the computational fluid dynamics. This is only for the upper airways, not for the lung or alveoli. So once the particle, like the nano-sized particle, the fragmentation of the micro-sized particle, or this is the alveoli, then how those particles will interact with the lungs affected molecular, that will be explained in this presentation. So this is how the airways is not a smooth out. That's why they have so many barrier and make some vortices. That can be done by using the console or ANSYS by the finite element and finite difference method. So these are the 500,000 of particle that then goes down in the G plan. So this is the source of micro or nanoplastics that we have from the personal care products, the plastics products as well as the textile. So that is the structure of those. It could be the polystyrene, polyolefin, polyethyl glycol, PVC, and so on. So how those nanoparticles, when you inhale that, it actually is to the very deep of the lung, that is the alveoli of our lung. So how those particles actually enter into the cell, so that mechanism will be discussed here. So this is the scale that I have used in my study. So that is the common source. There's the definition as well, micro nanoplastics. And this is the exposure route that can be used to develop my model. So this is the consequence, that how those nanoplastic or microplastic actually contaminate our or toxic our cell and interact the cell and also cause the cell death. So in the present study actually show the presence of the nanomicroplastic, not how they interact, how they penetrate into the cell. This cannot be identified yet experimentally. So my work is actually analyze those, how this nanoplastic actually enter into the cell. So before going further, I need to talk about how those cell membrane, that is the lungs affected molecule, are actually treated. So once we have alveoli, there are two types of cells, the pneumocyte type 1 cell and pneumocyte type 2 cell. So the type 2 cell actually secrete the alveolar bodies. When it reaches the aorta interface in our alveoli, then it will form a thin layer. That thin layer is known as the lungs affected monolayer. Down this monolayer, we have the bilayer and cell layer as well. So that is the schematic of the cell layer. Here we have different types of lipid, proteins, and other organelles. So this is the composition of those organelles, and that is the structure, chemical structure of those. We have designed those chemical structure in our modeling. So this is how we have designed our. Once we inhale the whole air, then it contains some nanoplastic or microplastics. In case of the nanoplastics, it will be deposited in the upper side of the lung. That calculation is done by just CFD calculations because it actually obeys the continuum properties of the fluid dynamics. But when the tiny particle, it doesn't follow the continuum properties. That's why the fluid dynamics actually fail to calculate those trajectories and other things. So then we need to introduce new dynamics, which is known as the molecular dynamics. So this is the alveoli. So I will work in only here, and that calculation will be done in the next study. Once the nanoplastics enter into the cell, and how the nanoplastic interact with the lipids of the lungs affected molecule. So the mechanism will be discussed in this study. So we can also answer those two questions once we have completed this study. So as you know, since in title I have used the mechanical things. So the air, when we